welcome to this explainer. Today, we are exploring a truly fascinating corner of the plant kingdom. Have you ever looked at a delicate leafy fern and thought, ah, yes, the snake of the plant world? Probably not, right? But that is exactly the dramatic nickname biologists have given to the group of plants we're going to look at today, the Pteridophytes. So why are they called the botanical snakes? Well, to understand that, we actually have to look back at their evolutionary predecessors, the bryophytes. Now, bryophytes are often called the botanical amphibians because they never really severed their ties to water. But the pteridophytes evolved right after them. Just like reptiles were the first vertebrates to truly conquer the land, pteridophytes were the first true terrestrial plants to develop sophisticated internal plumbing. They evolved vascular tissues, specifically xylem and phloem, which literally allowed them to pull water from the soil and distribute nutrients throughout their entire bodies. All right, let's take a quick look at our roadmap for today. We'll start by meeting the botanical snakes. Check out their habitats, zoom in on sporophylls, strobili and structure, walk through the prothallus and their complex life cycle, and wrap up with heterospory and some massive evolutionary leaps. Section 1. Meet the botanical snakes. Let's get right into what these plants actually are and why they matter. The core distinction between the botanical amphibians, the bryophytes, and our botanical snakes comes down to a major, game-changing evolutionary upgrade. Unlike bryophytes, pteridophytes possess true vascular tissues. Because they have xylem for water transport and phloem for nutrient transport, but still reproducing using spores rather than visible seeds, scientists classify them as vascular cryptogams, which just means their reproduction is hidden. They are absolutely the true pioneers of plant plumbing. So what do these pioneers actually look like? Well, the pteridophytes you're most likely to recognize are the horsetails and the ferns. And they aren't just evolutionary relics. They're incredibly useful to us right now. We use them for medicinal purposes, and their intricate root systems make them excellent soil binders, which helps prevent erosion. Plus, because of their lush, intricate leaves, we frequently grow them as ornamentals to brighten up our homes and gardens. Moving right along to Section 2, Habitats of the Pteridophytes. What is truly fascinating here is the sheer adaptability of these plants. If you picture a fern, you probably picture it in a cool, damp, shady forest, right? And it's true. That is exactly where you'll find the vast majority of general ferns. But they are way more versatile than they get credit for. Some species flourish perfectly well in sandy soil conditions. Genera like Salvinia, Azolla, and Ceratopteris have actually gone backward, returning to the water as true aquatic ferns. On the total opposite end of the spectrum, some species of Selaginella and Adiantum are xerophytes, meaning they're adapted to survive in incredibly dry environments. And then you have Marsilia, which is the ultimate generalist. It can occur as a terrestrial plant, an amphibious plant, or a fully aquatic one. Section 3. Sporophylls, strobili, and structure. Let's zoom in a bit and see how these highly adapted physical bodies are put together.
When you look at a fern or a horsetail, you are looking at the sporophyte. This is the dominant main plant body of the pteridophyte, and thanks to that vascular upgrade we discussed earlier, this body is differentiated into true roots, true stems, and true leaves, with all of these organs possessing well-differentiated vascular tissues. Now notice the distinct leaf sizes. Pteridophyte leaves come in two main varieties. They can be tiny microphylls, which are small leaves like those found on Selaginella, or they can be macrophylls, which are the large, familiar, sweeping fronds we see on typical ferns.
This brings us to a really crucial piece of terminology, the sporophyll. Sporophylls are essentially leaf-like appendages on the sporophyte. Their entire job is to subtend or support the sporangia. Think of the sporangia as these vital biological factories where spore mother cells undergo meiosis to actually produce spores. This right here is the very beginning of the next generation. And this brilliantly illustrates how plant structures can specialize. You see, in some pteridophytes, those spore-bearing leaves, the sporophylls, don't just set loosely on the stem. Instead, they tightly pack together to form distinct, compact structures called strobili, or cones. If you look at plants from the genus Selaginella or Equisetum, which is the horsetail, you'll clearly see these cone-like strobili perched right at the tips of their stems. All right, Section 4, the prothallus and complex life cycle. We've seen how spores are made, but what happens next is surprisingly dramatic. Let's look at the hero's journey of a single spore, step by step. First, as we learned, the sporangia produce spores via meiosis. Once released, if a spore lands in a hospitable spot, it germinates. But, and this is wild, it doesn't immediately grow into a new leafy fern. Instead, it grows into an entirely different, independent entity called a prothallus. On this prothallus, specific sex organs, the antheridia and archegonia, mature. Next, water physically transports male cells, the antherozoids, to the female archegonium. Finally, they fuse to create a zygote, which then sprouts into the multicellular, well-differentiated sporophyte we actually recognize.
I really want to pause on the prothallus for a second because it's just fascinating. This phase of the plant is officially a gametophyte. It's an inconspicuous, small, multicellular, free-living, and mostly photosynthetic little organism. It is entirely separate from the large parent fern that dropped the spore. But here's the catch. To grow and survive, this delicate little prothallus absolutely requires cool, damp, shady places. Let's look a little closer at the, let's call them romantic requirements happening directly on that tiny prothallus. It bears two sex organs. The male organs are the antheridia, which are completely embedded within the prothallus. They release male gametes called antherozoids, which are amazing little structures. They're uninucleate and spirally coiled, featuring flagella to help them move. The female organs are the archegonia, which are only partially embedded and contain the egg. Now, for fertilization to happen, those spirally coiled antherozoids literally have to swim from the antheridia to the mouth of the archegonia.
And honestly, that need to swim is the pteridophytes' Achilles heel. Let me read you a quote directly from the source material that sums this up perfectly. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. This is exactly why these botanical snakes don't completely rule the world anymore. Without a layer of water for the antherozoids to swim through, the entire life cycle just halts. Which leads us perfectly into Section 5, Heterospory and Evolutionary Leaps. I want to pose a question to you. If a single type of spore does the job perfectly well for most ferns, why do some specific pteridophytes go through all the trouble of producing two entirely different types of spores? Well, the answer lies in evolutionary innovation. The vast majority of pteridophytes are what we call homosporous, meaning all the spores they produce are of a similar kind. But a few rebel genera, like Selaginella and Salvania, are heterosporous. This means they produce two distinct kinds of spores, macrospores, which are large, and microspores, which are small. So the crucial point here is what actually happens to these two different spores. The large megaspores specifically give rise to female gametophytes, while the tiny microspores give rise to male gametophytes. But here is the monumental shift, the real game changer. Instead of dropping the spore to grow a vulnerable, independent prothallus on the damp ground, these heterosporous plants retain the female gametophyte right on the parent sporophyte for variable periods. When fertilization occurs, the zygote develops into a young embryo completely protected within that retained female gametophyte. By retaining the female gametophyte and developing the embryo directly on the parent plant, Selaginella and Salvinia achieved something absolutely incredible. This biological event is officially considered the evolutionary precursor to the seed habit. It was a monumental step in plant evolution, literally paving the way for the protective seeds that dominate modern agriculture and our global food supplies today. Which leaves us with this final thought. How did a simple change in the size of a spore, a split into macro and micro, triggered an evolutionary cascade that ultimately led to the seed-bearing plants, the modern forests, and the agriculture we rely on today? The next time you walk past a humble fern or a delicate stalk of horsetail, I hope you remember that you aren't just looking at a pretty ornamental plant. You are looking at a botanical snake, a pioneer of the ancient terrestrial world, whose evolutionary innovations shaped the earth as we know it. Thank you so much for joining me for this explainer, and keep questioning the incredible systems at work and the natural world around us.